Welcome everyone to our Meslium Center seminar series, which today is co-hosted by the Department of Radiology. And it's a real pleasure for me to introduce our seminar speaker today, Dr. Hwamee Oh, who is Associate Professor of Psychiatry and Human Behavior and Associate Professor of Cognitive, Linguistic, and Psychological Sciences at Brown University. She's also affiliated with the Carney Institute for Brain Science at Brown. Dr. O received her PhD in biopsychology at the State University of New York at Stony Brook, where she concentrated in human cognitive neuroscience. After she graduated, Dr. O did her postdoctoral training with Dr. Bill Jagist in the Helen Wills Neuroscience Institute at UC Berkeley. There, she studied alterations in preclinical older adults with beta amyloid deposition by using amyloid PET, structural MRI, functional MRI, and neuropsychological testing. Prior to Brown, Dr. O was an assistant professor at Columbia University and a research assistant professor at SUNY Stony Brook. At Brown, by using multimodal imaging, neuroimaging, Dr. O has extended her research to cognitive and neural changes due to normal aging and Alzheimer's disease pathology. And this is to understand the factors that are responsible for both individual differences in people and also how these factors contribute to brain aging and the relationship to cognition. Dr. O has several honors and awards, including the DeLeon Prize in Neuroimaging for New Investigators. She also received first place for the Alzheimer's Association Award for Excellence in Research on Alzheimer's and Related Disorders. She is active, very active in teaching and training at Brown and has numerous other roles both inside and outside of Brown. So without further ado, I'm going to hand the mic over to Dr. O, whose seminar title is Developing Novel Behavioral and Neuroimaging Markers of Preclinical Alzheimer's Disease. Dr. O.
Thank you, Dr. Foster, for kind introduction. And I also like to thank the Meslium Center for Cognitive Neurology and Alzheimer's Disease and the Department of Radiology for inviting me to visit here and present our work. It's been very exciting to see the ongoing research at the Meslium Center and across the Northwestern community. So my lab's research focuses on understanding the cognitive and neural changes in association with brain aging and Alzheimer's disease pathologies, with the primary focus of early detection of Alzheimer's disease. We believe this work will eventually help us to develop early cognitive and neuroimaging markers that advances early diagnosis, prevention, and treatment monitoring of Alzheimer's disease. So today's talk, I'm going to start with talking about the Alzheimer's disease and Alzheimer's disease pathologies and the current definition of Alzheimer's disease for the audience who may not be familiar with these terminologies. Then I'm going to review our work that examines the impact of beta amyloid deposition on cognition, brain structure, and brain function in preclinical Alzheimer's disease. Then I'm going to discuss the distinctive topographic distributions of tau neurofibrillary tangles and beta amyloid deposition in preclinical Alzheimer's disease to lay a ground for the work that we are currently focusing on. Then I'm going to talk about the two projects that aim to develop early cognitive and neuroimaging markers of a preclinical Alzheimer's disease and the way to harmonize different tau PET tracers. Then I'm gonna highlight the importance of individual difference factors that contribute to the development of Alzheimer's disease pathologies and cognitive impairment. And I'm going to share some thoughts on the next steps that that is needed in our research field. So this figure shows the projected number of people who is age 65 and older in the US with Alzheimer's disease, grouped by a different age range. And currently it is estimated about 6.9 million people, which corresponds to 1 in 9 people aged 65 and older suffer from Alzheimer's disease in the United States. And it is projected, the number is projected to double by the year of 2050. So you can sort of calculate, depending on what your age is, the higher likely the developing Alzheimer's disease. So it is very crucial to stop this projection. Alzheimer's disease is currently diagnosed based on clinical and neuropsychological assessments. But once the patient is diagnosed, they undergo a progressive decline, including the loss of functional independence and behavioral problems over an average of 8 years. Currently, the diagnosis to assist the diagnosis of Alzheimer's disease, the most frequently used neuroimaging modalities in clinics are FDG PET and structural MRI. With the FDG PET, the AD patients typically show hypometabolism in temporal parietal cortices compared to completely normal older individuals and patients with other dementia, such as frontotemporal dementia. In structural MRI image, AD patients typically show enlarged ventricle or sulci and severe hippocampal atrophy compared to normal older adults. The definitive, although the Alzheimer's disease is diagnosed based on clinical and neuropsychological evaluations, the definitive diagnosis of Alzheimer's disease has been done based on neuropathological evaluation and autopsy. And senile plaques consist of beta amyloid peptides and tau protein neurofibrillary tangles are considered as primary Alzheimer's disease pathologies. Although other types of proteinopathies, such as alpha-synuclein and TDP-43, are also commonly present in Alzheimer's disease patients, and which is the case more so as the disease of Alzheimer's disease progresses. So it is important to consider that there are several pathological changes co-present in AD Alzheimer's disease, and those may be collectively contributing to the cognitive and behavioral functional impairment. These are the pathological changes could be assessed by autopsy until recently. However, now we do have the ability to image these pathological changes in vivo. And these images are the amyloid PET using the tracer called Pittsburgh compound B from Alzheimer's disease patients, as well as cognitively normal older adults. And the warm-colored regions are showing where the amyloid plaques are deposited. There are many ways to classify individuals into amyloid positive and amyloid negative. But one of the methods that is commonly used is the kind of quantitative methods. And one of those quantitative methods is to use the amyloid plaque level observed in young adults who is about 20 to 30 years old, and derive the cutoff values. And we classify the individuals, whether they are amyloid positive, if they are in the above cutoff, or the amyloid negative in the below the cutoff. And the cyan colors represent in this plot showing the Alzheimer's disease patients, and you can see a majority of them are classified as amyloid positive, whereas the red colors represents the country normal older individuals, but you can still see the large proportion of country normal older adults are classified as amyloid positive. Approximately 30% of country normal older adults who are 60 years old or older are currently presenting the amyloid deposition detected by PET. And individuals who are country normal but presenting with amyloid positivity in the brain, then they are considered as conceptualized as preclinical Alzheimer's disease. These are the amyloid index, which I'm going to be repeatedly throughout this talk. So I wanted to give some kind of a background how those measures are sort of derived and represent what they represent. So, as a quantitative method, a simplified quantification method is frequently used using graphical analysis and distribution volume ratio or standardized uptake value ratio values are derived. In the autopsy data, the posterior singular gyrus that is highlighted here typically show high level of amyloid plaques compared to other brain regions, particularly in AD patients compared to control normal older adults. And when those PET signal coming from that regions are plotted in this time activity curve, AD patients show the increased, the higher binding signal throughout the scan compared to the currently normal older individuals. When we get this time activity curve from the brain regions, such as the cerebellum, then we don't see the difference in the PET signal between AD patients and control subjects, which is consistent with what we see in autopsy data. So in order to derive the quantitative measures, those PET signal in the across the brain regions are divided by the so-called reference region, for which we commonly use using the cerebellum, gray matter cerebellum, which gives us a ratio of the amyloid load, accounting for any types of individual differences in cerebral blood flow measures. So, which gives us a sort of normalized value of amyloid PET signal in a quantitative manner. And throughout this talk, I'm going to use amyloid index or global amyloid index, or the depending on the tracer, I may use the PIB index to indicate those aggregated cortical amyloid deposits. It is also important to know that amyloid deposition is highly related to age. So typically the amyloid accumulation begins to show at the age of 50 and 55, and it steeply increases at the age of 80. As shown in this meta-analysis, where each circle represents one study with a mean age of that sample in that study on the x-axis, and the proportion of amyloid positive individuals in that sample on the y-axis. So in the animal literature, there was a lot of studies showing significant impact of amyloid, beta amyloid plaques or proteins on cognition and neural systems. So, but there was a consensus that there is no behavioral differences in currently older adults as a function of amyloid pathology. So my early work examined whether there was any impact of amyloid pathology on cognition and brain structure and brain function, particularly in preclinical Alzheimer's disease. So first we examined recruiting the subjects, 44 young subjects and 52 older adults, currently normal older adults from the Berkeley Aging Cohort Study at UC Berkeley, and examined whether there is any difference in cognitive measures that we derived using different cognitive measures. And other than, among these currently normal older adults, other than amyloid index, there was no significant difference in MMSE scores or ApoE for genotypes, even the intracranial volume. Across these 5 cognitive domains, we found significant effect of age in visual memory and executive functions, but there was no significant cognitive difference in older adults as a function of amyloid positive and negative. But as you can see, the amyloid positive older individuals tend to show lower behavioral performance, cognitive performance across the different cognitive domains. So we aggregated these behavior measures across these 5 cognitive domains then we observed not only the age-related differences, but also A-beta-related differences. So this study is one of the first studies that actually examine the cognitive differences as a function of amyloid deposits among cognitively normal older adults. But we also was wondering whether these results may be the results of a bit biased sample bias that reflects highly educated older adults in the Bay Area in Northern California. So we conducted a meta-analysis by including different types of amyloid measures, including CSF measure, plasma measure, histopathology, PIB PET, and other PET measures. And across the different cognitive domains, we found that there is a significant effect of amyloid with lower cognition in episodic memory, executive function, and global function measures. Which is indicated by the effect size significantly different from 0. Also, the effect size of episodic memory was also significantly greater than other cognitive domains. This more studies, more meta-analysis on the same topic with a much larger number of papers were subsequently published by other groups and reporting the A-beta related differences in cognition with moderate effect across different cognitive domains, including global cognitive function as well as memory. So it's quite clear that across the different studies, we begin to see there is a cognitive differences among cognitive normal older adults as a function of A-beta pathology. So we also wanted to see whether there is any neural differences that may be underlie these cognitive differences as a function of amyloid pathology. So we look at the gray matter atrophy while we are controlling for chronological age, but in relation to amyloid index. With age, chronological age, we observed very substantial gray matter atrophy throughout the brain. But also among amyloid positive group, we found there was a significant brain regions that showed gray matter atrophy with a higher amyloid index, including the lateral frontal cortex and lateral parietal cingulate cortex and retrosplenial cortex. Since there are several neurodegeneration related markers are co-present at neuronal level, we further examined whether there is it's been difficult to say whether this atrophy was uniquely associated with amyloid pathology. So in this study across the Alzheimer's disease spectrum using ADNI data set, we found there was a lower CSF A-beta, which corresponds to higher level of brain amyloid plaques, are uniquely associated with greater cortical atrophy in multiple brain regions, as depicted in warm-colored regions, controlling for other CSF measures, such as CSF neurofilament light chain and p-tau 181, and demographic information accounted for. In the animal literature, there was also a lot of studies pointing that there is a different proportion of neurons that is either hyperactive or silent in APP transgenic mice. So we was wondering whether there would be any the neurofunctional differences in terms of the neuronal activity as a function of amyloid pathology. So in order to examine those functional aspects, we conducted fMRI study. In this study, both young and older adults view the images presented one by one on the screen, and they had to indicate whether there is water or not. So, and then after the scan, they were given the recognition task where they had to indicate whether they have seen this image during the scanning or not, and also how confident their responses were. So this study design allows us to examine brain activation patterns that occurring while they are viewing the images that may be later recalled correctly or not. And if the images are correctly recalled later, we consider that the encoding of the visual information was successful. First, we look at the brain activation pattern, the differences between young and confident normal older adults who doesn't have amyloid pathology. And we found the increased brain activation pattern in young adults than amyloid negative or confident normal older adults in the brain region that is highly implicated in visual episodic memory. But more interestingly, we found when we compared amyloid positive and amyloid negative older adults in this successful memory encoding activation pattern, we found there was an increased brain activation with amyloid positive older adults than amyloid negative older adults, including the frontal cortex and hippocampus. We also assessed whether there is any functional connectivity differences between amyloid positive and amyloid negative older individuals. In order to do so, we look at the functional connectivity with the right parohippocampal gyrus, which is highly implicated in visual episodic memory and the rest of the brain. And we found there was a brain region that show significantly decreased functional connectivity with a right parohippocampal gyrus in A-beta positive older individuals in these warm-colored regions, including right frontal cortex, superior parietal regions, and lateral occipital regions. So in this fMRI study, both in regional activation and functional connectivity collectively suggest that with beta amyloid deposition, there is increases in regional activation with amyloid, but decreased functional connectivity across brain regions. We examined this amyloid plaques and neuronal activation patterns using several different cognitive paradigms, including verbal working memory, executive control and function paradigm, and visual details memory paradigm, encoding paradigm in different study population, in study cohorts, and repeatedly found that there was an increased regional activation with beta amyloid in amyloid positive older individuals than amyloid negative older individuals. But it's also important to know that the A-beta related increases in brain activation during task is not infinite. It seems when the A-beta positive individuals show increased activation when the task is easier, but the activation compared to amyloid negative older individuals, but they tend to show the reduced brain activation compared to amyloid negative older individuals when the task becomes more challenging or difficult. So we interpret this as kind of the harbinger of the sort of subsequent cognitive decline with amyloid pathology. So I mentioned several studies that examine the impact of amyloid pathology on cognition and neural systems. But as the tau neurofibrillary tangles, tau pathology is also co-present in many cases with beta amyloid pathology. It is important to look at both beta amyloid pathology and tau pathology to understand their unique contribution to cognitive impairment and neural changes. So I'm going to talk about the distinctive topographic characteristics of distribution of tau neurofibrillary tangles and beta amyloid deposition in preclinical Alzheimer's disease as a kind of the background of the project that we move forward. It's long known that beta amyloid pathology and neurofibrillary tangles are co-present. and appear to develop, proliferate in a very predictably systematic manner, but topographically distinctive. So neurofibrillary tangles begin to show in the brainstem and entorhinal cortices, and then spread to the associating cortices at a later stage. Whereas beta amyloid, the abnormal aggregation of beta amyloid pathology begin to show in the associating cortices, gradually develop in those regions, and then move to the hippocampus and the sensory motor cortices. So it's been hypothesized that the amyloid pathology appearing in the associating cortices is mark the end of the tau pathology being restricted in the medial temporal lobe, and then spread to spreading to the outside of the medial temporal lobe. So now we can measure both amyloid and tau pathology using in vivo PET, and this is the tau PET imaging images shown on this the top panels using the flow tau sphere PET. And the first three images are coming from consciously normal older adults. And the next two images are coming from the patients with MCI, mild cognitive impairment. And the last two images are coming from the AD patients. So this, the pattern of the image that we see here, and then the clinical status is well fit with what we know from the clinical pathological data. It is also important to note that except the first subjects, the PIB index, which represents the amyloid index, are all amyloid positive, which is also consistent with what we know from the preclinical Alzheimer's disease. Especially the early stage of the AD pathology of amyloid and tau PET, there is a poor correlation in terms of the distribution and concentration between two pathologies. So in this participant's image of the PET scans, the amyloid PET scans using flow beta peer tracer showing high binding in the lateral frontal and medial frontal regions, as well as the temporal and parietal regions, whereas the signal is very devoid in the medial temporal lobe. The tau PET signal, tau PET data, on the other hand, show the opposite pattern of the binding, showing that there is a signal in the medial temporal lobe, but outside of the medial temporal lobe, there is no tau PET binding within the brain. So this, the topographic dissociation gives us a unique opportunity to examine the impact of each pathology on cognition and brain structure and function. The topographic distribution of each pathology is better, can be seen in this video. So the middle images are the tau PET signal from AD patients, and the rightmost images are from the amyloid PET signal from AD patients from the posterior to the anterior brain. And the warm colored, red colored regions indicating higher binding of each tracer. So based on this distinctive topography dissociation of amyloid and tau PET signal, we are currently conducting two projects aimed to develop the early cognitive and neuroimaging markers of a preclinical Alzheimer's disease. So one study is Brown multimodal imaging of cognition, aging, and Alzheimer's disease study, as known as Brown-Mieke study. And the other study is longitudinal multicenter head-to-head harmonization of tau PET tracers, as known as HEAD study. The preclinical Alzheimer's disease, as I mentioned, is the stage where amyloid pathologies are present, mostly in the associating cortices, while the tau pathology are being shown in the medial temporal regions. So in this, we launched this Brickman study under the NIH support to systematically characterize differential effects of age and AD pathology on behavioral measures and neural substrates of cognition, and also to investigate distinctive effects of early amyloid and tau pathology on behavior and neuroimaging measures and their predictability of Alzheimer's disease. So in the Brickman study, the large group of older adults, currently normal older adults, and young participants are enrolled into the study, and all undergo the extensive neuropsychological test and the novel computerized task at 3 different time points over the course of the study. And all participants undergo several neuroimaging sequences, including structural MRI, task fMRI, resting state fMRI, DTI, ASL, locus coeruleus, and FLAIR scans. And all older individuals are scheduled for the amyloid PET and tau PET scans. And we also have the plasma AD biomarkers collected and ApoE for genotypes are determined. And we also collect the affective and other lifestyle measures to understand the individual differences in this pathological process. Currently, 162 participants were enrolled when I checked the last time, and 66% of participants are female, and all older adults are cognitively normal. The extensive neuropsychological assessment is done for the behavioral phenotypes using the National Alzheimer's Coordinating Center uniform data set measures. And amyloid PET is collected using fludarabine PET, and tau PET is collected using MK62 PET in the Rhode Island Hospital. For the structural and functional measures, we assess the several neural markers, including white matter integrity and resting state functional connectivity, and task-based brain activation patterns during task. And in collaboration with Dr. Haley Jacobs at Martino Center at Harvard, locus coeruleus integrity scans, which is the brain region that is known to develop the tau pathology, the early tau pathology. In the HEAD study, which led by the Dr. Tariq Pascal at University of Pittsburgh and Dr. Suzanne Baker at Lawrence Berkeley National Lab, aim to develop some methods that allows us to harmonize different tau PET tracers. So for the past several years, several tau PET tracers has been developed. All tracers are good in terms of binding with the postmortem data, but there are certain level of variability across tau PET tracers, which makes it a bit difficult to assess the clinical and in vivo pathological correlation. So currently, 660 people are enrolled and all undergo clinical assessment and MRI and blood draw. And the subjects are getting the amyloid PET scan and also different types of tau PET scan using MK6240 tracer, flow tau spear, and Roche compound and pyramidal compound. This is a study where we use the subset of participants to see how these tracers behave differently in terms of their discriminability performance across different diagnostic groups. So each voxels represent the area under the curve to differentiate the different diagnostic groups. And warm colors representing the better discriminability. As you can see, the MK6240 tracer does better job than flow tau spear tau PET scan when we discriminate AD participants compared to amyloid negative continuum older adults, and which is the case also with when we discriminate MCI patients, amyloid positive MCI patients compared to amyloid negative MCI participants. So there is certainly the variation in terms of their ability of that each PET tracer, tau PET tracer afford, but those needs to be sort of there. We need a method that actually can allow us to make them comparable with each other. So so far I've talked about the downstream pathway that may be associated with AD pathologies on cognition and brain structure and function. But I'm going to change the topic a little bit now to talk about the individual difference factors that may be contributing to the development of Alzheimer's disease pathologies and cognitive impairment, which may be more tied to the upstream path of the pathology. The lifestyle factors such as the education or diet has been considered as a resilience factor that may be moderating the relationship between pathology and cognition or clinical presentation. However, there seems to be also the direct impact of these lifestyle factors to the development of Alzheimer's disease pathologies. So we assess the lifetime cognitive activities that occurred in different time points across one's life and look at whether that lifetime cognitive activity level is associated with the level of amyloid pathology. And we found that with a higher level of lifetime cognitive activity that occurred over the lifespan, related with a lower level of amyloid pathology among cognitively normal older adults. This relationship was also shown in the whole brain voxel-wise analysis on the amyloid PET data, where the dark blue regions are showing greater amyloid pathology with lower lifetime cognitive activity. So there is seems to be a relationship with what has been done during the lifespan that may be impacting on the development of amyloid pathology. In order to assess that relationship more directly, the US pointer imaging is currently ongoing to assess the lifestyle intervention, including exercise and diet, on the level of amyloid pathology and tau pathology, which is so the US pointer imaging study is a 2-year longitudinal study that is led by Dr. Susan Lando at UC Berkeley, where we aim to assess whether the lifespan, 2-year lifespan intervention, lifestyle intervention such as exercise and diet, is going to show the level of amyloid pathology or tau pathology to be alleviated. So currently the 2-year scan, the follow-up scan data collection is almost complete. So we hope to be able to analyze the data to show this lifestyle intervention effect on these AD pathologies as well as other anatomical markers. But based on the baseline data, we found that amyloid positive older individuals tend to show the more tau pathology in entorhinal cortex and also the several cortical regions that was captured by meta region of interest compared to amyloid positive older individuals. Not only the lifestyle factors, but also the regional differences in the brain may be related to the development of amyloid pathology. In the animal literature, there was a lot of studies that's showing the amyloid pathology are regulated by the neuronal activity. So there is a higher neuronal activity, then there is a more production and secretion of A beta peptides. So we were wondering whether the regional differences in glucose metabolism that is observed in young adults are going to be able to explain the differences in amyloid pathology that is regionally a bit selective. So to do so, we used a free-surfer-based analysis to capture the multiple brain regions, and we get the FDG PET signal from those regions from both the hemisphere and plotted by the age groups. And based on the young subject's FDG level that is captured by this blue bar on these charts, we ranked all these regions in terms of their level of glucose metabolism and grouped the high quartile regions as high metabolic regions and lowest quartile regions as low metabolism regions. When we map those the region of interest on the surface data, these silver-colored regions capturing those high metabolic regions, and the orange-colored regions capturing the low metabolic regions. These metabolic differences within the brain was observed across the age range from young and older adults, but that glucose metabolic rate differences disappear in 80 participants, indicating that throughout the brain, there is a hypometabolism. Also, we found that in the high metabolic regions show the higher amyloid accumulation in amyloid positive older groups compared to the lowest FDG glucose metabolic regions. So collectively, the results suggest that the amyloid accumulation is not universal throughout the brain, but more selective, and there might be the regional characteristics of the brain that may be contributing to the development of amyloid pathology. And one potential mechanism might be the increased neural activity throughout one's life in those regions. These regional, relationship between the glucose metabolic rate of young subjects and amyloid accumulation in amyloid positive older individuals are also shown in this whole brain data. So amyloid accumulation is also, however, accumulates during the older age. So we was wondering whether there's any factor that may be explaining that the pathology is occurring during age. And we are particularly interested in the role of white matter aging in instigating amyloid pathology during older age. We examined the white matter integrity changes as a function of amyloid pathology across the different tracts, and we found that the tracts such as long-range tracts such as superior longitudinal fasciculus and corpus callosum show the lower reduced white matter integrity with associated with a higher amyloid pathology. So there are several explanations that why this white matter aging may be contributing to the regional differences in amyloid accumulation and also in individuals. So those are the areas that we are planning to look into further. Also, using the ADNI data set across the Alzheimer's disease spectrum, we also found a greater longitudinal reduction in white matter aging was associated with the subsequent amyloid plaque load across the Alzheimer's disease continuum. And this relationship was observed not only across the whole Alzheimer's disease spectrum, but also within each diagnostic groups. So last, I'm going to talk about where we are what area that needs to be further to be done based on what we found so far in terms of the effect of amyloid on cognition and Alzheimer's disease pathologies on cognition and on neural substrates. The traditional neuropsychological test has been done a great job in differentiating patients with mild cognitive impairment from cognitive normal older individuals, but may not be optimal for differentiating healthy aging from preclinical Alzheimer's disease, in part due to the very small effect size. And also, the task-based fMRI is good measures compared to resting state fMRI data to understand the functional changes in the process of Alzheimer's disease, but as it can be considered as kind of a stress test, and also it has a potential to be developed as a to improve more precision medicine by individual mapping. But the current task paradigms are very long, which can be seen very impractical and costly from the clinic perspective. So developing much shorter scan time is the paradigm that has can be collected with much shorter scan time is going to be important. In addition, the current way that we get the information about the presence of AD pathologies occurring in the brain is invasive, especially in the amyloid PET scans or CSF measures. So having the non-invasive and more practical, shorter, and digitally accessible or remotely accessible for the people who may not be able to come to the clinic often, and more sensitive measures to detect early and subtle cognitive and neural differences in preclinical Alzheimer's disease. And also, it will need to be responsive to the treatment where the field is more moving towards, and with a larger sample size. So in the MICA study, we are currently using and testing this paradigm using two-back task, where the long-term memory component is embedded and can be collected within about 10 minutes long inside of a scanner. And we are going to examine the behavioral measures coming from this task paradigm, as well as the neural activation pattern over the course of the 3 years. So far, I mentioned a lot of the measures that show the relationship with each other with AD pathology measures and brain structure and functional measures and cognitive measures, and also the more upstream measure that may be contributing to the development of AD pathologies. However, we also know the genetic components and lifestyle factors affecting these neuroimaging markers and behavior directly or moderate the relationship between these two measures across the measures. So in order to predict whether the individual has the pathology on the brain or will develop the cognitive impairment in the long run, needs some kind of integration approach using machine learning methods. So once this large data set is collected, we will work with the people to develop the way to integrate to have the better predictability. So I'd like to thank my lab, current and past lab members, and also the collaborators across the United States and also at Brown University. And thank you for your attention.
Open up for questions. We have mics for the people that are on Zoom.
Yes.
Marcel.
So you notice the correlations and relationships you saw. There was a huge range that the number of people who didn't seem to mind amyloid or tau was quite substantial, even though the mean may be different. So did you try to look at people with a lot of amyloid, but no change in cognition? A lot of tau, no change in cognition. What do these people look like?
Yeah, that's a great question. And Dr. Meslam, actually, I learned a lot about the importance of actually evaluating and studying at individual level. The results that I've been showing is based on the group data. So certainly that will need to be done, and I look forward to work on it. Yes.
Thank you. Thank you for a lovely talk. Just following up on that and the drive towards individualized measures of brain function, I was wondering if you could talk to us about the test reliability of some of these PET markers, because you have a high signal within a certain patch of cortex. How much should we believe that? At what level do you take that as true?
So in terms of a group level analysis for structural data is a bit easier to compare across the studies and quite typically showing the lateral frontal and parietal regions in retrosplenial and posterior cingulate cortex atrophy in relation to amyloid pathologies, which is quite well mapped, where actually also a higher level of amyloid pathology are shown. But in terms of the functional measures, is going to be a bit more task dependent in terms of where we are going to look at. So the retest reliability is going to be very important to justify and validate these functional functional fMRI-based measures. At least there are a lot of studies, I think that Dr. Braga know better than me, but there are also the studies that showing quite good retest reliability in terms of resting state fMRI data, in terms of the connectivity measures. So when we show some connectivity measures across the brain regions, either seed to seed measures, then that connectivity measure is quite repeated when the scan is done in the different time for one participant. So subject-based wise, quite robust with the functional resting state functional connectivity data. But in terms of the task-based data, I think that is important question, and less answers are currently available.
So my question was specifically about the PET images. So, you know, even when you're measuring atrophy with the structural MRI, there is error there that we normally don't well capture. Some new approaches are showing that if you collect multiple T1s, you can actually reduce the error term. Is there something like that for PET, or will it be difficult to associate that with atrophy and fMRI?
PET data wise, I think that there is a quite good test reliability, but in terms of the correlation with structural measures, I don't can think of any kind of a study that shows within the study, but I can say across the studies, those kind of the correlation that might be reflected in where we see the atrophy in relation to PET amyloid index are quite consistently reported in the same brain regions. So in that sense, there is a certain the reliability in terms of that. But I think your question is more about how reliable this PET signal may be when we scan the participants two times, and then maybe impacting on how what we observe in relation to other neuroimaging markers. So yes, I think I do not see the kind of I cannot recall any study that show that relationship in that manner. But in terms of the already PET signal, as long as we use the same tracer, it's quite reliable in terms of the PET signal. But when we change the tracer, the pattern is going to be slightly changed because of the different characteristics in terms of the offsite of target binding.
Thank you.
I have a question. So, Hwamee, on one of your slides, you showed the spatial paradox between the development of amyloid pathology and the development of tau pathology in different regions. They're just showing up in different regions of the brain with a different time course. Can you speculate about that spatial paradox, and can your work on A beta positive, tau negative preclinical individuals speak to understanding that spatial paradox?
First of all, I don't think anyone at this point knows why the tau start to accumulate in entorhinal cortices, and amyloid pathology begin to appear in association cortices. So that we still don't know. But with the amyloid accumulation, a lot of people actually also develop the tau pathology to a certain extent. So people think that when there's no amyloid pathology, tau pathology is more restricted within the medial temporal lobe, which is actually shown in the autopsy data, even in the subjects about age teens or 20s. So it is not age related, but for somehow those regions have the tau pathology accumulated in those regions. But it is restricted in normal controls. But once the person has amyloid pathology develop in the brain, they're sort of instigating those tau pathology that used to be restricted within the medial temporal lobe as part of brain aging, goes out, spread outside of the medial temporal lobe. And people think that amyloid pathology may be sort of expedite or accelerate some kind of hyperactivity. They maybe also have these medial temporal lobe tau proteins spread through the synapses to the other regions. That's how the amyloid pathology may be influencing the tau pathology's development. In the preclinical stage, still the tau pathology is restricted within the medial temporal lobe, but it's going to spread out throughout the other parts of the brain, starting within the inferior temporal regions and posterior cingulate regions that is connected to the medial temporal lobe.
Thank you for your talk. I was just wondering if you can reliably classify someone as preclinical AD individual or normal AD individual solely based on A beta accumulation.
I'm sorry, would you repeat your question again?
Yeah, sure. I can also rephrase. So if we use amyloid beta accumulation as the only marker, where do you draw the line between normal aging and preclinical Alzheimer's?
Okay, thank you. So for that's a great question. Whether it also touches upon how we are going to be interpreting the amyloid pathology, whether it's going to be considered as part of normal aging, or it's going to be considered as the pathological aging, not the normal aging. Current understanding about the amyloid pathology is considered as not normal process, normal brain aging process, more is considered as a pathological brain aging process based on the genetic the studies that are linked to the amyloid pathology. But I think that's a great question, but in the field at this point, amyloid pathology is considered as part of the pathological aging.
Tracy, do we have any online questions? Okay. Well, we're about out of time, so I want to thank. Oh wait, Borna.
Thank you for the beautiful talk. I found it very fascinating that there was an association between lifetime cognitive engagement and low amyloid. Based on your experience or your work or other people's work, how much do you think amyloid or tau imaging is sensitive on picking up interventions like that. The study you mentioned, I think that was lifelong cognitive engagement. How about, in the clinic, we recommend people exercise, we could recommend cognitive stimulation. Is this something that we can pick up on amyloid or tau imaging?
Yeah, that's a great question. In terms of the amyloid, we in normal controls, we see the change in amyloid index even a year and a half. So using the global quantitative measures, so it is feasible to see whether that change is going to be affected by 2-year intervention. We just don't know whether that 2-year intervention is going to be effective to that. In terms of the tau pathology, it's a bit open question. Tau, if it is under in the medial temporal lobe, then probably the accumulation rate is going to be not as strong as the amyloid pathology. Yeah, I think that's a great question.
All right. Well, thank you, everybody. This was a great talk. Thank you, Hwamee.